Sure. Before I begin, let me just briefly introduce myself for those of you who are meeting for the first time. My name is Daye Lee, and I'm a senior bioconsultant at Rokit Healthcare. To describe our company in one sentence, we are an organ regeneration platform company that focuses on translating 3D bioprinting technologies to the next level. So we provide 3D bioprinters and the relevant systems to not only research groups, but also, I guess, in the long run, we envision providing them to doctors for treating patients at point of care. So with that said, because we just have 1 hour today, I'll go straight into descriptions of our systems. And then the rest about our company, the background, I can just share with you guys in a corporate profile.
Yeah, no, I'll share with them. But no, you can share your screen now. I allow you to, yeah.
Okay.
Perfect. We got you.
So, yeah, I've just described a bit about our company. By the way, if I go back here, so the philosophy behind creating our bioprinting systems, the biggest philosophy is the fact that these will eventually go into the hospital market to change the way we do healthcare. So we named our systems in vivo, meaning that everything that's made biofabricated from these systems would ultimately be for in vivo applications. And there are two systems that we provide. One sort of in the medium price range in the market, if I just talk about market terms, and then the other one a bit more in the medium premium versions. The first one is called the Dr. In Vivo 4D. And this is what the system looks like, and we will get to see more pictures of it later on. The key thing about Dr. In Vivo 4D is that it is the world's first clean chamber topped bioprinter, meaning it was the world's first to introduce HEPA filter, the HEPA H14 filter, and the UV germicidal lamps built inside the bioprinter. And the idea was that if you are printing cells or biological systems, these would be contained within a clean chamber. Now, it is also a hybrid bioprinter, which means that it can do multiple types of materials. These materials range from hydrogels with or without cells. That's why it's in its essence called a bioprinter, because it can print cells. And then it can also do thermoplastics, such as filaments, pellets, and powder. Here are some examples of the materials we can print. With the syringe biodispenser, you can print hydrogels such as collagen, gelatin, chitosan, alginate, etc. With the extruder, filament extruder, and the hot melt dispenser, you can print materials like PLA, PLGA, hydroxyapatite, even polystyrene and polyurethane. The temperatures are important here. For any bioprinter, the coverage of different temperatures is very important. So you can actually create printable materials within this temperature range. So the temperature range that we can cover includes -4 degrees to 60 degrees for the syringe biodispenser, room temperature to 250 degrees for the filament extruder, and room temperature to 350 for the hot melt. The key thing is the in vivo is able to go below 0 and above 250. These are the competitive advantages of in vivo over other products in the market. And the cool thing is, due to a need that arose in one of our user bases, we actually also created this customized optional bed platform that you can install onto the bed, and you could put dry ice under it. And what that does is you can get the bed's temperature even more below zero, down to -20 degrees. And you would have the advantage of printing certain hydrogels, such as alginate, which print better, or actually not print better, but which stabilize better under very, very cold temperature. So at Rokit Healthcare, Dr. In Vivo, the bioprinter, is being used for a variety of R&D applications. So our company is not just a supplier of bioprinting products, but we also do our own R&D, with each of the teams responsible for different projects. And these projects range from dermatology to orthopedics, ophthalmology, cardiology, and development of human cell-based cosmeceuticals, cosmetic and pharmaceuticals. But before I go deeper into those applications, I'm gonna go a little bit deeper into the hardware systems. So I gave you a brief overview of what the system is about, but let me just go a bit more in detail. So if you look at Dr. In Vivo, it is cylindrically shaped. It's the only bioprinter out there that is cylindrically shaped. And there was an aesthetic purpose here, but the other purpose is that we were concerned about, with any 3D printer, there is a concern of vibrations intrinsic to the printing process that could potentially affect the resolution of the printing or the stability of the structure that you printed. So the cylindrical design allows it to at least absorb or evenly spread out any vibrations that happen during the process. And in addition to that, if you take a look at the hardware, inside there are these little accessories that make your printing process streamlined. And one of them is the thermometer that monitors the chamber temperature. You have this filament bobbin that allows your thermoplastic filaments to stay in place as you are printing. And finally, there's this bio-ink warmer. It's these two little holes that you can put the syringe dispensers in, and they can remain at a stable temperature while you're not using them. So here are these little accessories that help you, use your system with ease of use. So I've talked about the temperature of the dispensers, the hardware design overall, and the little accessories inside. And I will talk a bit about the things that help you cross-link your materials after printing. So in bioprinting, other than the thermoplastics that we are used to in 3D printing, people use hydrogels a lot. And the great thing about hydrogels is that they tend to be biocompatible, and they are soft enough to encapsulate cells. And so you can print cells directly onto some type of bed, right? But the thing about hydrogels is that often they may be printable, but they may not necessarily be able to hold their shape after you print them. They could crumble once you have printed them. So to help with that, there are hydrogels developed so that they can be cross-linked and become hardened upon exposure to certain environmental factors, such as temperature, UV or light source, and chemicals. So our system is capable of doing all three of them. Temperature, as I said before, the syringe dispensers the syringe dispensers and the bed can be controlled in temperature from -4 degrees to 60 degrees. And these are enough to affect hydrogels that are temperature cross-linked. We also have UV lamps at 365 nanometers and by default, and customized wavelengths if you wish, so that certain hydrogels can be cross-linked when you shine that UV light on them. And these UV lights are located at the bottom of each syringe dispenser. Sorry. But at the end of the day, the most important things of every bioprinter are the dispensers that allow you to print. So I will briefly summarize these dispensers one last time, and then I'm going to move on to the next system, the next generation system that came after this. So the hot melt, I told you that there are three types of printheads Dr. Invivo provides. One is a syringe dispenser, a filament extruder, a hot melt pneumatic dispenser. This hot melt pneumatic dispenser can accommodate nozzle sizes of 100 microns to 600 microns, and it is specialized for pellet and powder printing, and especially high viscosity material printing. It can go up to 900 kPa of pressure to dispense a variety of materials. Its operating temperature range is, as I said, up to 350 degrees, and the types of materials you can print are outlined below. And it would look something like this, where you put your pellets inside a stainless steel barrel, and this barrel goes into the printhead. The filament extruder is a type that all of you who may already have experience with FDM 3D printing would be used to. This is a filament extruder that can do nozzle size from 100 to 400 microns, and the operating temperature range is 250. And as long as your materials are provided in filament formats with 1.75 millimeter diameters, you can potentially accommodate onto this printhead. And the most commonly used ones are PCL, PLA, PLGA. And this is what it looks like in real life picture. And finally, the syringe dispenser accommodates from 100 to 600 microns. These are just nozzle sizes that are replaceable, nozzles that are replaceable. So potentially you can use any variety. It's just a matter of, can you print your materials at the pressure that this can provide. Oh, I think there's a typo here. Please disregard this. This is for low viscosity materials printing with hydrogels. And the operating temperature range is -5 to 60 degrees. And the types of materials it can provide, it can print, include a variety of hydrogels from collagen, gelatin, alginate, hyaluronic acid, etc. So I'll correct these and give them to you. And this is what the syringe dispenser looks like. And finally, the bed. The print bed dictates the volume in which you can print, and it also dictates the speed at which you can print. The build volume is 10 by 10 by 8 centimeters, and this is pretty standard to the bioprinter market today, especially of our price range. And the printing speed, it can go up to 20 millimeters per second. And the operating temperature range is also -5 to 60 degrees. This is -5. But comfortably, people often use -4 to 60 degrees. And so if you can combine these different configuration, like I said, the printer itself is a 2-axis printer, meaning it can use 2 materials in one contract. But because we have 3 different printheads, you can have more than 2 combinations. So you can have a combination of hot melt with syringe for some type of hard tissue and soft tissue combinations, filament extruder and syringe dispensers, as well as syringe and syringe. So this gives you the types of combinations you can expect from Invivo 4D. And with Dr. Invivo 4D, it comes with these external controllers that are sort of the dedicated powerhouses of our system. The reason that ours can go down to -4 degrees or -5 degrees for the bed and the syringe dispenser is because of the temperature controller that has coolants inside, and this is unique to Dr. In Vivo. Also, the reason that the hot melt pneumatic dispenser can go up to 9 bars, 900 kPa of pressure, is because of this air compressor system that actually goes up to 11 bars at maximum at its operating pressure. So these external controllers are what give Dr. In Vivo 4D the relatively high specifications that it has for temperature as well as pressure. And these are just the summary of that. The key thing is that because different groups have different budgets and different groups also have different research needs, so we do provide Dr. In Vivo 4D in different configurations. The things that I just explained are all part of the premium, the highest spec. But if you go down to upgrade and standard, you take out certain specifications. So if you go down from premium to upgrade, you remove the hot melt pneumatic dispenser. If you go down from upgrade to standard, you remove any type of temperature regulation on the syringe dispenser and the bed. Okay? And it comes with its own slicing software that can create, that can input 3 STL files and convert them into instructions for the printer. And like I said, comes in different configurations for budget-specific and research-specific needs. And finally, with Dr. In Vivo 4D, some accessories we can provide include this human cell-derived whole ECM material. It can come in supplements like powders that you can add to your own material, or it can also come in hydrogel format. And we also provide in vivo gel, which is basically a variety of gelatin-based biomaterials with or without growth factors for tissue-specific applications. So with that, I will conclude the presentation on the Dr. In Vivo 4D. And I spent a lot of time on this just to describe sort of the fundamentals of bioprinting and what each of the components means for a bioprinter. But I'll just spend 5 minutes on the next generation bioprinter. And maybe if you have questions, I'll try to address them during the last 30 minutes. Okay, so that's 4D. Now, let me just show you something. Let me show you. Okay. Before I transition to 46, we have had 4D, Dr. In Vivo 4D for about 2.5 years. First, sort of establishing the market here in Korea, which is where our headquarters is based, and expanding to the global market starting in the mid-2018. And over the last two to 2.5 years, we have established about these many user bases, about 250, of which we have 15 publications on the use of Dr. In Vivo 4D, and there are 5 clinical/preclinical case studies. And these case studies include the development of customized bone implants for patients in the hospital, the development of heart patch, the development of a heart patch for ischemic heart disease modeling, as well as for transplants. And there's the application of rhinoplasty, meaning the development of PCL scaffolds embedded with the patient's own fat tissue to insert into their nose for rhinoplasty surgery. And there's the application of wound healing in diabetic foot ulcer patients, and finally, cartilage regeneration. And I won't go into too much detail today, but as you can see, the application areas range widely across various fields of tissue engineering and regenerative medicine, because the bioprinter is just a platform. You can just think of it as a box, a black box, and if you include the cells, the biomaterials, and the 3D design of your interest, the output will always be different, right? So the applications are varied, but not just in tissue engineering and regenerative medicine. There's a lot of research going on in material science, just characterization of new materials, mechanical engineering, engineering of new devices, ergonomic devices, pharmaceutical sciences for the development of novel pills, and also food science, like food printing, meat printing. And finally, even in veterinary medicine, which is like regenerative medicine for humans, veterinary medicine for animals. So having said that, after all these applications that have been developed, although they are ongoing, what we have been learning is that there are still needs, there are still challenges in bioprinting that we need to resolve in order to advance to the next level of tissue engineering and regenerative medicine. So, yeah. So what we learned is that, okay, it's a good step forward to include the HEPA filter and the UV lamps to create this clean chamber environment, but it's not enough, especially if you imagine it going into the operating room. It needs to be able to do more so that whatever is printed inside would be really effective for transplantation into the patient, right? So we identified 3 key requirements for real tissue and organ regeneration. And these are also the feedback of researchers and doctors that we met. The first requirement is the idea of in situ printing. So if you can print not just on planar surfaces, but if you can print on curved surfaces, because the body itself is a curved surface, that would be important for creating in situ direct applications of bioprinting for wound healing right away. Number 2 is a requirement of sterilization. Originally, I said that UV and HEPA filter are enough for disinfection, but they're not enough for complete 100% sterilization. So if you can imagine, biofabricating something inside the printer and implanting it into the human body, you need this sterile environment. And finally, perhaps the most important thing of all, we think, is the idea of cell incubation. Mammalian cell cultures, they know where they grow best, right, at their native conditions, comprising of temperature, in vivo pH, CO2 tensions, and humidity. If you can control these environments inside the bioprinter, wouldn't you expect higher cell viability and better conserved phenotypes of these cells as they are getting printed? So we combined all of these 3 into one. And the result is called Dr. In Vivo 4D6. Let me skip over these and show you some videos. So we combine these three into one, and the result is Dr. In Vivo 4D6. The 4D6 includes a CO2 incubation chamber of its own. So you can control the temperature, maintain it at 37, maintain humidity at 95, and CO2 level at 5%. And the idea is to protect cells during printing and after printing from any environmental change shock. As an option, you can also include a plasma sterilizer of low temperature H2O2. And this basically sprays H2O2 liquids into gas plasma particles that completely sterilize the chamber as well as the material inside if you wanted to. The Dr. In Vivo 46 is equipped, just as the name suggests, it is equipped with 6 printheads total. You have 5 printheads that are in a rotary biodispenser format. So you can print 5 hydrogels at once on the rotary dispenser. And then on the left-hand side, you have a lone unit of a dispenser that can be interchanged with 3 different types. And these 3 different types I already talked about before in the 4D. They're filament extruder and hot melt pneumatic dispenser. And the great thing about 46, there have been many advancements in 46 from 4D, right? There's the inclusion of a cell incubator, sterilizer, and the increase of printheads from 2 to 6. But also, it has been equipped with the highly precise linear robot motor. This is something you don't usually find in a bioprinter, which is often just done in stepper motors or belts. But we have included a linear robot that you might find in a medical device. So with these all in mind, this is a video of how the printer operates. So you have 5 syringe dispensers rotating to create a complex architecture. To create a complex architecture with different biomaterials and bio inks. So the more complex you can get, the better you can get in a tissue engineering project, right? Next, you have the temperature, CO2 humidity level control. So our expectation is better cell viability and cell functionality after printing. And the HEPA filter is still conserved here. It hasn't been lost. And with 46, you can do high throughput applications up to 384 wells. That opens doors to a tremendous amount of opportunities in high throughput drug screening, high throughput toxicity test development, vaccine research, and even PCR genotyping if you need to use microliter volumes in automated ways. So, and you can do this all in one in one bioprinter. With the use of these 3 different printhead technologies at up to 6 printheads in one construct, you can print as complex of a tissue or biological system as you would like, with a variety of materials. And part of this list includes the FDA approved materials such as PCL, certain types of alginate and gelatin and fibrin. So if you can imagine using the bioprinter for medical commercialization, that would also be possible. I mentioned about putting the bioprinter in the operating room by Rokit Healthcare last year to treat diabetic foot ulcer patients in India. But in 2020, we've also seen our partner company called BioOrigin that has developed a GMP process for creating customized bone implants. So this company went from a biomaterial company to a customized bone production company using in vivo in a GMP process. So I think this is an excellent example of a company sort of opening a new business model using the bioprinter.
I have a question real quick.
Yep.
So number one, when you're saying about soft tissue, when you're talking about soft tissue for diabetic foot ulcer, you guys are running a trial in India, I understand. So
we have finished trials in India. Yep.
oh, you finished already. Okay. So you're using biomaterials and you're using also autologous cells, or you're using autologous cells?
Yes. Autologous solutions that are taken from adipose tissue, from liposuction. And then, yeah, these are made into a customized patch using the bioprinter. So there is no culturing involved. It's all point of care, meaning that the autologous solutions printed in a customized patch get applied to the diabetic foot ulcer right away. And then you expect healing over the course of the next several weeks. And we have seen on average 4 weeks to 8 weeks of healing.
So they're basically debriding the ulcer, and at the same time they harvest some adipose tissue, and you do the printing, and it's placed on the wound immediately.
Yes.
And so with the customized bone implant, you're not using cells, you're using all biomaterials.
Yeah, all biomaterials. These are, I don't know the exact details of the materials because they're proprietary to this company, but they include things like hydroxyapatite and PLLA. And I think they also developed their own bone material that like mineralizes faster than just the natural materials that they used before.
Now, do you have something on the dermatology besides the wound care area? And I'm more interested in clinical applications because I want to see the practical. I believe that's the basics of what we're trying to do, like clinical applications. So besides the wound care, you said something about dermatology. Are you treating any other conditions with that, or or aesthetic
currently
something?
Yeah, currently this is a project of our R&D team here. That's called the skin regeneration team. The focus indication right now is on diabetic foot ulcers, but we do see potential applications branching out to burns and scar removal. Yeah. Also, I guess, like, you know how I talked about rhinoplasty applications with the inclusion of biomaterials inside the nose? Maybe after you do that, you would also need some type of healing on the skin here. So it could be used for in parallel with the rhinoplasty applications as well, I think.
Am I there? Am I there? Not maybe? Are you hearing me?
Yes.
Yeah, we can hear you.
Oh yeah, well, my Zoom, I don't know what happened to it. It just got flat.
I hear you.
Are you practicing doctor in dermatology or?
No, I do mostly regenerative medicine. And basically we do some wound care.
Mm-hmm.
That's why I was really interested in that. Jorge, I don't know if you have any other, like that you could see any other application?
Traumatology,
Trauma, yeah.
yes, and urology, maybe.
And all that will be using adipose. You will be printing the material and injecting it during surgery, and then based on adipose, right? You're not using allogenic cells at all?